Thank you. Thank you also for the introduction, Antonios, and thanks for having me. So I'll jump right into it. Um, so I think animal models are very important for studying tinnitus because with animals we can take tissue samples from the cochlea or from the brain, which is very hard to do in humans, and we need to do that to really understand the cellular mechanisms and molecular mechanisms of tinnitus. But what makes animal studies difficult is that we don't have a ground truth measure to really for sure say like that animal has tinnitus and that one doesn't. So because of that, any changes we see in the animals, we're often not that sure whether it is due to hearing loss or actually due to tinnitus. So I think if we want to better disentangle the pathophysiology of hearing loss and tinnitus, we need to have translatable tinnitus biomarkers that we can apply both in humans as well as in animals. And in this study, we're trying to take a step towards that. So one of the mechanisms that we're trying to work with is the central gain mechanism. So with tinnitus, we often see loss of hair cells and synaptopathy in the cochlea. And that, of course, leads to decreased activity in the cochlear nerve because there's less input. But centrally, we're seeing more signs of hyperactivity. So that could be a compensatory response to the hearing loss. So if tinnitus is a side effect of too much central gain, then by measuring central gain, perhaps we can also infer tinnitus. So with our animal model, we're working with genetically identical animals. And then we do noise exposure on a subset of them. And then we see that all of them get hearing loss. And now we want to know which ones of them have tinnitus, or if they all have tinnitus, which ones has more tinnitus than the other to kind of look specifically at the tinnitus-related changes. And to do that, we're applying three different measures. So the first is using the auditory brainstem response. So here we're looking at the amplitude of wave 5, which corresponds to activity of the inferior colliculus, and compare it to the activity of wave 1, the activity of the cochlear nerve. So we're kind of looking at the central response to a sound stimulus relative to the peripheral input as a measure of central gain. The second method is using GPIS, which is the most commonly used behavioral assays to assay to detect tinnitus. And here you play a background sound that may sound like tinnitus, followed by a startle stimulus. And if you proceed the startle stimulus by a silent gap, then you see that the startle response gets inhibited. But after noise exposure, we see that many animals don't have this gap-induced inhibition of the startle response anymore. So that could be due to tinnitus-related changes in the processing of the frequency of the background sound, that there is hyperactivity around the tonotopic areas of those frequencies, and that is masking the ability to detect those gaps. And the third method is using a biomarker that we've identified in humans. So this is from a study that's currently running at our department. And here we're seeing increased GABA A receptor binding potential in the primary auditory cortex of tinnitus patients, even when controlling for hearing loss. So you can see that this finding has very high predictive power of tinnitus. So we want to try to take this to the animal model to see if we can replicate it and to look at the mechanisms underlying this change. But also perhaps to validate the other behavioral and the electrophysiological measures of tinnitus that we're using. And we're doing that here with this measure. We're taking PET scans with a drug called flumazenil, which binds to the GABA A receptor. So we want to cross-compare these three measures with one another. So to do that, we've used 32 mice, 5 are in a control group and 9 are in 3 noise exposure groups. And we want to make sure that we have enough noise exposure to actually induce tinnitus. But we don't want to have so much noise exposure that the animals have too much hearing loss that they cannot hear the stimuli anymore from GPIS and the auditory brainstem response. So that's why we're trying different amounts of noise exposure. So we did 3 baseline GPIS measurements and baseline ABR. And then after noise exposure, we did 9 weekly GPIS measurements and 2 post-exposure ABRs, which were 2 months apart. And then we did PET scans in a subset of the animals to look at the GABA A receptor binding potential, and then collected the brains and cochlea of all the animals. Okay, so let's jump into the results. So we have the hearing threshold on the y-axis and the frequency on the x-axis. You can see that there's a very nice dose-response relationship between the amount of noise exposure and the amount of hearing loss. And we can also see that the phenotype is already pretty stable just 2 weeks after noise exposure, because 9 weeks later, there's not much further changes. So what about central gain? So let's take a look at this representative trace from a control animal. And here we see a relatively large wave 1 and a relatively small wave 5. And we see that with more noise exposure, this trend tends to reverse. So that wave 1 gets smaller, which makes sense because you have less input going through the cochlear nerve with the acoustic damage. But wave 5 either remains about the same size or gets even larger. So this could be the central compensation. And we see that in most noise-exposed animals, but interestingly, not all of them. So here we have an animal with a small wave 1 and a very large wave 5. But this animal has had the same amount of noise exposure and also has a small wave 1. But here, wave 5 remains quite small as well. And we took these ABRs twice after noise exposure, and they remained the same for these animals for both ABRs. So we're also seeing here that on a group level, the more noise exposure you have, the smaller the wave 1. Whereas for wave 5, so the activity of the inferior colliculus remains relatively preserved at the higher stimulus intensities for the 110 and 115 dB noise exposure groups. And at 120 dB noise exposure, the animals were practically deaf. So if we express this 5:1 amplitude as a ratio, then we can also see significant increases in the ratios in these two noise exposure groups. And if tinnitus is a side effect of too much central gain, then it may be that these animals up top here have more severe tinnitus than the animals down below here. So what about GPIS? So if we look at the 110 decibel noise exposed groups, we can see that the startle amplitude with a gap is smaller than without a gap. So that means that these animals can still detect the gap and it's not getting filled in, so to speak, by tinnitus. At 115 decibels, we can see that this ratio of the startle amplitude with and without a gap right after noise exposure shoots up towards 1 and stays there. So that means that there's no more change in the startle amplitude with and without a gap. And if we look at some individual animals here, you can see that indeed now the startle amplitude with and without the gap is the same because these lines are perfectly overlapping. But this animal still has intact gap detection. So it looks like some animals are getting behavioral evidence of tinnitus according to GPIS and some don't with 115 dB. And at the 120 dB noise exposure, we know that these animals cannot hear the background sound anymore because if we replace the background sound and gap by just a noise pre-pulse, then that also doesn't induce inhibition anymore. So that means that these animals cannot hear the background sound anymore. So the question is, does this line up with the ABR data? So we're still analyzing the data, but it looks like the animals that have intact gap detection are not the ones that also have lower central compensation according to the ABR. What about the PET data? So the GABA A receptor binding potential seems to correlate with both hearing loss as well as the wave 5:1 ratio we measured with the ABR, but not the GPIS ratio. But it's important to note that we've only done PET scans in 8 animals, so these correlations are insignificant. And you can also see in the raw data here of the PET scans that the data is very noisy. And that makes sense because we're working with these very small mouse brains. And in humans, the areas are just much larger, so you just get a better resolution. So I think we need to do some more work to really be confidently able to say whether the GABA A receptor binding potential correlates with either any of these measures. But for now, we found that noise exposure of 110 and also 115 dB results in increased wave 5 amplitudes relative to the wave 1. So that could indicate central compensation to hearing loss. And then maybe that then the animals with more gain also have more severe tinnitus. We also see that most of the 115 dB exposed animals have gap detection deficits at 12.5 kilohertz, which could be due to tinnitus. But we don't see these deficits in 110 dB exposed mice. We can't measure it in 120 dB exposed mice. Lastly, we also perhaps see a correlation between the GABA A receptor binding potential that we measured with the PET scans and both hearing loss as well as central compensation, which could reflect tinnitus-related changes in GABAergic signaling. But we need to do some more work. So the next steps are to perform ultraradiography using the brains of the animals in this study. So we're taking slices of the brains, incubating them with flumazenil, this GABA A binding drug that's also radioactive. And then we let the radioactivity shine onto a screen to get these super high resolution images of the binding of flumazenil to the GABA A receptors. And this is what such an image looks like in a mouse brain. And I think you can appreciate that this resolution is much higher than in PET. So then we want to calculate the binding potential again and compare it again with GPIS and the central compensation measured with the ABR. And hopefully we see some of these measures line up, that we have some more confidence that we're actually measuring tinnitus. And otherwise, we'll just proceed with using the GABA A binding potential as our primary classification tool. And then for the next experiment, we're going to apply spatial transcriptomics. So we're going to look at the gene expression in different areas of the auditory system. So the inferior colliculus, the medial geniculate nucleus of the thalamus, and the primary auditory cortex, and then compare mice with both hearing loss and high tinnitus to mice with hearing loss and either no tinnitus or low tinnitus. And then hopefully we get a better understanding of why there are changes in the binding potential of flumazenil to the GABA A receptor, but also get a better sense of the molecular processes that are happening in tinnitus as a whole, so that we can continue investigating them further, and hopefully this will also inform new treatments in the future. So that's where we are right now. Thank you for your attention.